So allow me to make an illustration. Imagine someone jumping from rock to rock along the shores of California. We can see them jump further and further away into the water. Now, would it be wise to conclude that because we can observe them jump in the direction of Hawaii, that, therefore, it's possible to jump all the way to Hawaii? Of course not. You might be jumping in the right direction, but the problem is that there aren't rocks you can jump to all the way to Hawaii. There is no step-by-step pathway. Now let's compare that to evolution. In order for evolution to work, things must evolve one step at a time. As Darwin said, if it could be demonstrated that any complex organ existed which could not possibly have formed by numerous successive slight modifications, my theory would absolutely break down. As you know, these changes happen at the molecular level, and in our illustration, a mutation is likened to a person jumping. Most of the directions you jump in are detrimental, and you'll fall in the water. But some mutations occur that happen to convey an advantage. This is like jumping from one rock and landing on the next rock. We'll compare this to a single-step mutation. Evolution can work great when explaining these types of small, single-step changes. However, the problem arises when two specific mutations are required in order to convey an advantage. This is like coming to a rock that's twice as far away. It'll be much more difficult to jump there. If the next rock is 3 or 4 steps away, the jumper is out of luck. He simply has no step-by-step pathway to get there. Now, the same is true for any beneficial advantage that requires 2 or more specific mutations. If a change requires 1 mutation, great, evolution can explain that. But if 2 or more mutations are needed, the mechanism of evolution starts to lose its power. As Alan Orr acknowledged, given realistically low mutation rates, double mutants will be so rare that adaptation is essentially constrained to surveying and substituting 1 mutational step neighbors. Thus, if a double mutant sequence is favorable, but all single amino acid mutants are deleterious, adaptation will generally not proceed.
As I explained in a previous video, most mutations are neutral or nearly neutral. In the quote OFNF just read, Alan Orr was only talking about beneficial mutations. He did not say that evolution can't work if multiple mutations are needed to convey a selective advantage. Neutral mutations can be spread through a population by genetic drift, which could then allow further mutations to occur which are beneficial but would previously have been deleterious. To understand what I mean, let's look at biologist Joe Thornton's research. Thornton studied the evolution of steroid hormone receptors. He wanted to know how the glucocorticoid receptor or GR and the mineralocorticoid receptor or MR evolved from a single ancestral receptor. The GR is activated by cortisol and the MR is activated by aldosterone in tetrapods and by deoxycorticosterone or DOC in fish. The scientists worked out what the most likely amino acid sequence of the ancestral protein that existed 450 million years ago was by sequencing animals alive today. They then made this protein in the laboratory. The receptor protein that they made was sensitive to both cortisol and aldosterone. Since aldosterone first evolved in tetrapods, this means that animals had a receptor that could be activated by aldosterone before it even evolved. The gene for this ancestral receptor was duplicated and each copy evolved different specificities. The scientists reconstructed the series of mutations which led to the changes in specificity. They found that certain mutations had little or no effect on the activity of the receptor but increased its stability. These mutations would have had little or no effect on the organism's fitness and so would not have been favored by natural selection. These mutations could have been fixed in the population by genetic drift. Without these neutral mutations, beneficial mutations which occurred later would not have been possible because they would have destabilized the receptor and made it non-functional. You may say that this exact series of mutations was extremely unlikely to happen in the order necessary, and I would agree. Just like it is extremely unlikely that a specific person will win the lottery. That does not mean that it is extremely unlikely for somebody to win because somebody has to win eventually. Evolution has no goal. The ancestral receptor was not trying to evolve into the modern receptors. If that series of mutations had not happened, a different series of mutations would have happened. Instead of the receptors we have today, the ancestral receptor might have evolved into different receptors. The research on the evolution of steroid hormone receptors shows that just because multiple mutations had to happen to convey a selective advantage, it does not mean that these mutations had to occur simultaneously in a single individual.
So here we have a limit to what evolution can do. Now, of course, there's a few ways around that to help your odds. such as increasing the mutation rate, as HIV does. Another way is to have astronomical population size, again, as HIV does. This is obviously impossible for larger animals, but for single-cell organisms, it's not as much of a problem. In fact, Oxford University malariologist Nicholas White noted that in any 2-day period, ill people could contain between 5 times 10 to the 6 and 5 times 10 to the 7 malaria parasites. These provide extremely large populations, yet even with these massive populations, here's what he admitted. Resistance to chloroquine has arisen spontaneously less than 10 times in the past 50 years. This suggests that the per-parasite probability of developing resistance de novo is on the order of 1 in 10 to the 20th parasite multiplications. So we can ask an interesting question, one that concordance never stops to consider. Why does it require such large amounts of parasites for chloroquine resistance when it takes far fewer to counter other malarial drugs? What's the hurdle to chloroquine resistance? Why does evolution have such a difficult time evolving this? Well, Michael Behe predicted this in his book Edge of Evolution back in 2007, and he was actually just recently confirmed in 2014 in PNAS by Summers et al., who wrote that, quote, the minimum requirement for low chloroquine transport activity is 2 mutations, end quote.
Resistance to chloroquine in the malaria parasite is caused by mutations in a membrane transport protein known as PFCRT. These mutations enable the protein to pump chloroquine out of the cell's digestive vacuole. In the quote OFNF read, the authors say that 2 mutations had to occur to enable low transport activity. They did not say that these 2 mutations had to occur simultaneously in a single individual. In Figure 3 of the paper, the authors show that there are multiple mutational routes to effective chloroquine resistance. The green arrows represent an increase in chloroquine transport activity, while the red arrows show a decrease and the blue arrows show no change. As you can see, there are 2 possible first steps, both of which are neutral. This is why it requires 2 mutations to enable low transport activity. The first mutation could be spread throughout a population by genetic drift and allow a second beneficial mutation to occur later in a different individual.
And so it's not difficult to understand why, when multiple mutations are required for a change, the likelihood of it happening drops exponentially for each required step. That's why chloroquine resistance was so difficult to overcome, despite extremely high mutation rates and an extremely large population. Again, 2 things generally not available to any mammals or birds, fish, or reptiles. So here's the thing. We can both agree that random mutations can accomplish some things, single mutation steps, and even double mutation steps if we have enough trials. Beyond that, evolution loses its power. In 2008, some critics tried to refute the intelligent design proponent and biochemist Dr. Michael Behe in the journal Genetics. Rick Durrett and Dina Schmidt wrote an article titled Waiting for 2 Mutations with Applications to Regulatory Sequence Evolution and the Limits of Darwinian Evolution. They failed to refute Behe, noting that in the human population, to obtain only 2 simultaneous mutations via Darwinian evolution, it would take more than 100 million years, which even they admitted was very unlikely to occur on a reasonable timescale.
In the paper, OFNF is citing the authors were calculating the probability of 2 specific mutations occurring simultaneously in a single individual. Evolution is not trying to achieve specific mutational goals. The first mammals were not trying to evolve into humans. If they had not evolved into humans, they would have evolved into something else. If the series of mutations which led to humans had not happened, a different series of mutations would have happened. Given that the average human inherits about 77 mutations from their parents, it would be entirely possible for 2 mutations to occur simultaneously to accomplish something that would not be possible by single mutational steps.
Even the outspoken atheist Jerry Coyne has confirmed that it is indeed true that natural selection cannot build any feature in which intermediate steps do not confer a net benefit on the organism.
In the quote OFNF just read, Jerry Coyne did not say that every single mutation had to convey a selective advantage. He said that natural selection could only build features where the intermediate steps were beneficial. As I have already shown, genetic drift can fix neutral mutations in the population, allowing further beneficial mutations to occur which would not previously have been possible.
But based on the research that Dr. Michael Behe has done, it seems that natural selection has the power to overcome even a 2-step mutation in certain cases. Scientifically, that seems to be the limit to what evolution can do. You may be able to jump from one rock to the next, even if there's a step missing in between, but you'll never get to another island by this process. Now, although you haven't considered the implications of this, I think you at least understand the principle on an intuitive level when you said the following.
It's much harder for the virus to evolve a resistance to drugs targeting replication than it does for them to evolve new mechanisms of entry.
So here you acknowledge that some things are more difficult to evolve than others. With this in mind, isn't it odd and scientifically reckless for you to then want to extrapolate this and try and account for all the diversity of life on Earth? If a virus has a hard time finding a new mechanism of entry, what makes you think a single cell can turn into a giraffe? Is it even possible in your mind that there might be some constraints somewhere along that biochemical pathway? If you're having a hard time jumping along the rocks on the shore, why would you believe you can make it to a new island?
A single cell turns into a giraffe all the time. It happens whenever a mommy giraffe and a daddy giraffe make a baby giraffe. Scientists do not extrapolate the changes we see in organisms today to the entire history of life. The reason they have concluded that giraffes and every other living thing is descended from a common ancestor is because of the evidence from fields such as comparative anatomy, genetics, molecular biology, paleontology, embryology, and biogeography. I think that covers all of your arguments then. I know the possibility is extremely remote, but I hope in the future you will be more careful about where you get your information from and that you will not make arguments that have already been made and refuted many times.